right, welcome to the second of our Unit 0 lessons. In this one, we're going to look at diagrams and graphs, how to read them, how to make them. And in case you are wondering what on earth this complicated diagram is, that's actually a diagram of all the metabolic reactions, all the chemical reactions happening in one of your cells. Yes, very scary. Luckily, you will not have to face something like that on the actual AP test. So, let's talk about graphing. So depending on whether the AP test is given in person or whether it's taken online like last year, you may or may not have to create graphs. And so what we need to talk about is how do you graph? And how do you graph appropriately so that you can earn maximum points? So just a note, this was just an example of a graph that I saw on the news one day, and I was just like, no, 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 that's bad science. What you'll notice is that there is no y-axis on this graph. And in fact, when I saw this on the television, I yelled out, where is the y-axis? And of course, there was no answer for me. And so if you look at this graph, it gives you a certain idea. It gives you an idea that cancer screening and prevention services are decreasing dramatically below abortion that have been offered by Planned Parenthood. However, when you have an actual y-axis and then you graph these accurately, you see a very different pattern. And note, I'm not trying to advance any political viewpoints. All I'm trying to say is graphs matter. Axes matter and units matter because without them, you can provide misleading information. If that is a drug trial or if it is a experiment trying to decide on life-saving treatments, that can cost lives. So, when you are about to graph, what you need to ask yourself are the following questions. What is the independent variable and what is the dependent variable? As you saw in our previous lesson, we have our good old friend DryMix to help us remember what axis we graph each of those on. Remember, the dry, the DRY stands for dependent, responsive, and then it's on the y-axis. And the mix part, the I stands for independent, and the X stands for x-axis. So that should help you remember where you graph each of those. Then you need to know what is the range of each variable, because you are going to be marking out potentially demarcations to show, okay, what is 5 days? What is 10 days on this graph? And if you are not careful, then you might accidentally mess up. And the problem is you are supposed to do your free response questions in pen, and you can't erase pen. That provides a bit of a problem. So you need to be able to graph accurately, you need to be able to graph effectively, and ideally be able to graph your data on the very first try. And then you need to know what information is the data giving you, because it's not so much on the AP test, but when you're in college, there are times when, depending on the set of data you're given, you may have to choose a different way of graphing. But we're not going to talk about that now. And then you need to ask yourself what type of graph would best show this information. Line graph, bar graph, histogram plot, there's all sorts of graphs. Mostly, you're going to be asked for a line graph or a bar graph. So if you have continuous data, meaning this is some variable that is changing across, like, let's say time. Let's say you're measuring the growth of plants. Well, if you're measuring the height of a plant, that is something that is changing constantly. So for example, if I had a graph of the height of a plant versus the time, I could have my data points. Okay. And then in theory, if I connect these with the line, I should be able to predict, oh, hey, at this time interval that I don't have on my graph, what should the height of the plant have been? On the other hand, if you're looking at something in discrete groups or sets, then you're going to use a bar graph. I have not seen any questions on the AP test which ask about having you draw a pie chart, so that should not be necessary. And then I'm going to put into your notebook files something called a graph choice chart. And what I'm going to show you with that, we'll look over it, is the fact that it's got all these suggestions on when to use what graph for what type of data. Now, luckily for y'all on the AP test, the vast majority of the time, they're going to tell you what type of graph they're looking for. Is it a line graph or is it a bar graph? You have to pay attention to this because I have had many students, both on tests in my class and some when grading the AP test, who were asked to provide a bar graph and instead drew a line graph. And even though everything else was correct, they weren't awarded any points for it. So be careful about this because you usually have at least 1 point on a graphing question come from actually plotting the data. And if you do the wrong type of graph and they've told you which type of graph they want, you're not going to earn that point. And every point counts. It could be the difference between a 2 and a 3, or a 3 and a 4, or even a 4 and a 5. So I do say to always include units, and I wanted to emphasize this. What does this graph mean? What type of data is it trying to show you? What results am I getting from it? Well, right now, all it really is a squiggly line on top of a grid. This data has no meaning because I don't know what it's graphing. You always need to include labels for your axes. So what quantity are you graphing on each axis? And you have to include the labels. Because if I just put, for example, that the x-axis is time, was it time measured in minutes, days, years? What's my unit of measurement? Because it's not going to, the purpose of graphs is to give meaning to your data and allow you to make predictions and describe trends. And if you don't know what you're graphing and you don't know how much you have in your units, you're not going to be able to draw any real meaning. And another thing is, usually on graphing questions, you will earn an entire point just by labeling your axes correctly. But while that seems like, oh, well, that's it, that's all you have to do to earn that point. It's a really easy point, and so many people miss it. So I'm gonna write a caveat here. You don't actually have to write a title on your graphs on the AP test. It is best practice to do that just because if you are, say, writing a research paper or a lab report, having a title for your graph is going to make it a lot easier for whoever's reading it to understand your data. But for the purposes of the AP test, you don't need to include that. You always need labels for your axes, including units. And then this is also a big problem. You need to scale and label your axes appropriately. And we will talk about that in a moment. When you're graphing multiple data sets, make sure each data set is labeled. So if you're graphing like with caffeine and without caffeine as your sets of data, make sure you have it clearly labeled. The best way to do that is if you have data points, to use different symbols for the data points, or if it's a line graph, you can use different lines. If you happen to have multiple colors, which you probably shouldn't, you could use different colors, but honestly, you're only supposed to write in black or dark blue ink, and we usually give the pens when you guys are testing in person. Another thing you need to keep in mind is that you will be given, if you have to graph, a set of axes, a grid, and you have to decide a lot of the times, okay, how much of this am I going to graph in? You want your data to fill as much of the area as possible, because if you don't, you've got all this wasted space. If you scrunch up your data so it's only in a tiny part of the graph, then you're not going to necessarily see any trends. So for example, if I had a set of data and I graphed it like this, I've got a whole bunch of empty space that I'm not using on that graph, and that's not very effective. Instead, what would be better is if I basically stretched out my axes so that way I can see what's happening in most of the data. So this is ideal, and I know that is a messed up check. I know it looks weird. So here's when I talk about scaling. So a few things are, well, is this correct? Notice I've got about even space between each of those numbers, meaning there's just as many lines or grids or squares, whatever you want to call it, between 2 and 7, 7 and 8, 8 and 20 and 25. Another thing I want to point out is that 2 looks like it's about in the middle right here. 7 looks like it's right on the line. 8 looks like it's in the middle. 20 looks like it's a little off to the side, and 25, those are just in kind of random spots. This is why I will always, always put tick marks on the side. So that way I can say, okay, this is 50, this is 40, this is 30, and so I'm very, very clearly marking where I have my numbers. So why is this not correct? Well, the tick marks are one thing, but another thing is, if you notice, there's equal distance on this graph between 2 and 7. That's a difference of 5. Whereas on this part of the graph, there's pretty much equal distance, but it's only a difference of 1. So it's going to skew your data. Basically, if you graph like this, where you only put the data points that you're measured on the graph, like if you only measured it 2 minutes, 7 minutes, 8 minutes, 20 minutes, and 25 minutes, and you put each of those, you spread them out evenly, all of your graphs will look like lines. If you don't believe me, try it. So now we have a um, different orientation. So I did forget to fix this. So what is different about this graph from the previous one? Well, one thing is the span measured between, like, by that label A is exactly the same as the span measured by that label B. So A looks like it's measuring about 2.5 days. B is also measuring 2.5 days. So equal distance on the graph is equal difference in whatever variable I'm looking at. And then I want to point out the data points that you're plotting, depending on the data you're given, may not be on one of these lines. So for example, I have 3 data points here. One of them is right at a nice intersection. That's fine. Another one is on one of the grid lines, but it's not on a horizontal grid line. And then this one right here is just smack dab in the center. That is still fine. You are not trying to make your graph look a certain way or look pretty. You're just trying to reflect the data. So if, for example, you had something that was like 17 days, well, you're going to graph it where it's 17 days. So that's a lot of graphing. We will try and do some graphs throughout the year. It's going to be more difficult when we're doing virtual learning, but we'll do our best. But now, last year's test was very, very heavy on this concept, interpreting biological models. So this is a very complicated model, not as complicated as that one on the first slide, but it's much more complicated than anything you'll see on the test. However, these kinds of diagrams are something you need to be able to interpret. So let's go over a few of the types of diagrams you'll see. So all of these images that I'm showing you right now come from previous AP tests from the free response sections. So some of these diagrams may show ecological processes. One of the easiest ones to see is, of course, ecological succession. And it's like showing you a picture of the species that are in an area over time. So notice it does have an axis. It's not quite a graph because it's a diagram, but it's fairly easy to read. The diagram that's on the left is a little weirder. It's more understandable in the context of the question, but you'll notice you have CO2 that you're starting with. You've got an arrow showing that that CO2 will then go into organic molecules, and then you've got another arrow showing that those organic molecules will then be converted into CO2. So hopefully you can kind of guess from this that this is a diagram of the carbon cycle, and this step right here is photosynthesis. And if you don't know that, you will by the end of the year. And this step right here is cellular respiration. And like I said, the context of the question helps a lot more. You may also have some diagrams that are showing physiological processes that happen inside the body. Um, you are less likely to see the one that's like the cross section of the spinal cord and skeletal muscle fiber, just because the nervous system is no longer covered on the test, but you still need to be able to interpret diagrams like this. A few things you need to pay attention to. Read all the labels, especially when they're pointing out the structures. Another thing, pay attention to the arrows. The arrows are usually either indicating the direction of movement or steps in some process. You see the same thing in this diagram of it's a reflex that they're basically showing. These arrows are showing that you have a stimulus. So you prick someone's finger. That stimulus is going to travel in this direction along this pathway. It's going to keep going in this direction. It's going to hit this junction where it's going to travel between nerves, hit this junction where it travels between nerves, and then it's going to come back out this way to a muscle. Now, you guys are probably not going to know all of these details off the top of your head because we're not going to be covering reflexes. But you should at least be able to tell basically that you've got information moving in a certain direction, and you should be able to trace the direction that information is moving. In the diagram on the left, you may not know what a neurotransmitter is. We'll learn. Um, but you should be able to realize that this circle, whatever it is, seems to be moving, and it moves down to this end of what they're calling the presynaptic neuron, and it looks like it opens up and releases those little bubbles out into the space outside. As we go through these and we start learning about more of these terms, they'll start to make more sense. But I will tell you, the AP test is really good at throwing some diagrams that I've never seen before on there. They'll show you different ways of recording data, of graphing data, and you've just got to kind of roll with it and be flexible and try to understand what you're looking at. Then you'll see some where there are cell processes. You may or may not get background information. I'm pretty sure in each of these you get some background information, although on the diagram on the left, you really needed to kind of read through it carefully and then look through the picture. So once again, you're going to pay attention to the direction of arrows. You're also going to pay attention to any labels that tell you where things are happening. So for example, this is showing you that you have a vacuole, okay? Then you have labels for each of the different molecules, and you also have these ions, and you have water. So in this question, it was very important to pay attention to things like, hey, I got an arrow going this way, and it's pointing out hydrogens. I got an arrow going this way, and it's pointing out potassiums. Also the fact that, hey, I've got some hydrogens moving this way, and I've got some potassiums moving this way. So it was very important in this question to pay attention to the direction of movement, the molecules that were allowing that movement, and so on. In the diagram on the right, this is something we'll learn about when we talk about DNA and a little more details about like transcription and translation. So you see you have DNA. The DNA is going to be used to make something called a primary transcript, which is then going to leave this kind of a bounded area, and it's going to go out into the rest of the cell, and it's going to be made into a protein. And from that, after we've gone through enough material, you'll be able to tell that, hey, this is the nucleus, and this is the cytoplasm, which can help you make some inferences that might be helpful on the question. This is what was very common on this past year's AP test, and it was biochemical pathways. We're going to look at a couple of examples. Here you see this is a pathway showing how one molecule is changed to another molecule, is changed to another molecule. So it's a pathway showing how the amino acid tryptophan is broken down and turned into these other molecules. You don't need to know what I3PA is. You don't need to know what IAA is. Instead, you need to know some details like the fact that, okay, we've got a gene, we make RNA, and then from that we get an enzyme. A lot of times, if you have a pathway that's showing enzymes performing a reaction, you'll see your molecules, and then you'll have the name of an enzyme. It's going to be over the arrow that's showing that chemical reaction happening. And then you'll have your next molecule, And then the next step in the pathway will have the name of its enzyme, whatever helps perform that step will be written above the arrows. So that's one thing to keep in mind that I will remind you of when we talk about enzymes. So like I said, arrows are pretty important. Of course, it can get more complicated. These were actually diagrams on a previous AP test, like all of these other questions, and this may look very scary, but this is something that we'll go over. We'll go over these diagrams, we'll go over what they mean, but you can still draw a few conclusions. So looking at the direction of the arrows, you can see in, for example, glycolysis and pyruvate oxidation, that you start out with something called glucose. How do I know you start out with it? Well, it's the beginning of the arrow, and there's nothing before it. My end products are anything that comes at the ends of the arrows. So these are end products, and then finally this. These are all end products, so I can see what's coming out of that reaction. So arrows are important. In figure 2, we're seeing something different. Anything that is coming out at the end of an arrow is going to be a product. So all of these things are getting produced. These two things are not getting produced. They're actually molecules in this cycle. But notice we have something coming in. So we're going to have two of those coming in every time we run through this cycle. And if you're like, I don't get that, we're going to talk about the Krebs cycle. We'll learn to love it. It lets you live. And then finally, this electron transport diagram, once again, is showing you, hey, this molecule, NADH, is going into this, and this is what we're getting out. Here, we're starting out with, they're saying half of an oxygen molecule, and our product is water. So you're seeing things that are produced. You're also seeing some of these diagrams are showing you movement. So they're showing you that, hey, hydrogen is being produced and hydrogen is moving. You're also seeing movement of basically these electrons. And one thing I would do is if in some of these diagrams, you're like, I can't trace quite where the arrow is going, feel free to draw extra arrowheads like I just did, because that helps you remember, oh, okay, all of this stuff is going to the right. Another type of diagram that you'll commonly see is a cell signaling pathway. This was on one of the last few years worth of AP tests. Not last year, because I don't know if I can show you those questions, so I'm not going to until I get otherwise information. But there was, of course, a very detailed description that went with this, describing what was happening. But once again, direction of arrows. Where is this starting? Where is this ending? Well, if you need to figure out where this starts, try and trace back the arrows. Where is an arrow starting? So this active interleukin right here, okay, an arrow starts there, but there are arrows pointing to it. Like for example, from this inactive interleukin and from this active caspase. Okay, the active caspase, does it have any arrows pointing to it? Oh, it's got this one right here with bacterial infection. So the starting point of this guy is probably going to be this inactive caspase because it is what triggers all the rest of these reactions. And then you can see that your end results are, oh, hey, this guy goes out because notice I don't have anything else that arrow is pointing to. So that's probably the target, the end reaction. And then I've got this guy going into the cell membrane. Now, when you read the descriptions, you'll be able to interpret this better. You'll be able to understand it a little more, but you can at least gather some information from these diagrams, even without the background info. And then finally, probably your second most favorite diagram that you'll run into on an AP test are things like phylogenetic trees. Most students that I've encountered over the years are not fans of the biochemistry parts of things, but they're great with evolution and ecology because those seem to be a lot easier for people to understand. And so phylogenetic trees are likely to show up on the test, although I will say that this past school year, because of COVID, we did not cover the units for evolution or for ecology. So there were no questions like this on the last year's exam. But this year, I anticipate all 8 units will show up on the exam. One thing I want to point out that you see in some graphs, you'll see these little hash marks or squiggly lines. What that means is they're basically skipping a bunch of time. It's like they took that axis and they took that section of it and they compressed it. So just keep that in mind. That happens sometimes. That's why you can have a jump from 40 hundreds of thousands of years ago, which would be basically like 4 million, I think, years ago, to 400,000 years ago. Apologies for the yawn. Now, those were diagrams that you'll see on the AP test. These are diagrams that you will see from our textbook, and I mostly concentrated on the biochem ones since they were so heavily emphasized this past AP test. But what you'll notice is kind of what I showed you on what I drew on a previous slide, where you start with the molecule and you start this pathway of reactions, a series of reactions. So here you start with molecule A, and the first reaction uses enzyme 1 to turn it into molecule B, which then undergoes the second reaction using enzyme 2 to turn it into molecule C, and so on. That's a very generic, showing you just kind of how enzyme biochemical pathways work. On the bottom, we have what's more like a series of chemical reactions. So here we have molecules, glutamic acid, and then ATP, which hopefully remember is the energy of the cell. And then what happens is a reaction that attaches a phosphate to this glutamic acid. And then you're going to have another chemical come into the reactions, ADP, and then you're going to add in this guy, an NH3, to make this molecule, and then those things, which can then go on to make more ATP. Obviously, the textbook part where I got this from has descriptions of what's going on, tells you the context, but you can at least tell a few things. What items are coming into this set of reactions, what is leaving, where the reactions are taking place, and the molecules involved. Then this is a bit more complicated of a diagram, but this is just showing how you have some chemical reactions that can happen in cells that are leading to some feedback. And here they're kind of color coding it, which you won't see on the AP test because it's in black and white. But the green means, just like what it does with the traffic light, go. So that step is going to take place, or it's going to encourage that step to take place. And then the red means stop. So what that means is when you have isoleucine, whatever this molecule is, it inhibits this enzyme. And so it is going to block this pathway. And that is something that I want to point out in this diagram. So I've told you to pay attention to where the arrows go. But what happens when it's not an arrow? What happens when it looks like this, or this, or this? When you have, instead of an arrow ending, you have that kind of line, that means it's inhibiting or blocking. When you have the arrowhead, that means it's making it happen. So keep in mind, when you have cells, there's always these complicated series of like some things are being inhibited or turned off, some things are being activated or turned on, and all of those things can kind of interact with each other. When you see that kind of block line, that horizontal line, that means it's blocking or stopping it. And then on the other hand, when you see an arrowhead, that means either a reaction is proceeding or this is where it's going. So those are all important things to pay attention to. So that's it for this lesson. We will, of course, go over many diagrams in class, as many as I can throw at y'all, and hopefully they will get easier to understand. So I am going to put a link in the description that leads you to a TED Ed video that I think is really good at explaining why it's important to graph things correctly and have accurate graphs. And then your fill in the blank notes will be in Google Classroom at another time. All right, I'll see you for the next lesson.